Good morning everyone. So today topic is C4 cycle are also called Hutch and Slack cycle. C4 cycle are also called Hutch and Slack cycle. So earlier we knew that first photosynthetic stable product was 3 phospho acid. Actually it is a product of C3 plants. C3 plant. But later observed by two scientists, they are Hutch and Slack in sugarcane and in maize, discovered that the first stable photosynthetic product was 4 carbon compound, that is OAA, oxalo acid. So that they found an alternative pathway, that is C4 cycle. In C4 name was came because of the first stable photosynthetic product is 4 carbon compound, that is oxaloacetic acid. And generally this C4 cycle occur in C4 plants are also called monocot or monocotyledon plants like sugarcane, maize and grasses and sorghum and millets. But there is a exception. The C4 cycle also occur in C3 plants like C3 plants also called dye plants like amaranthus and also in tribulus. Here this C4 cycle occur in two types of cells. They are in mesophyll cell and also occur in bundle sheath cell. And C4 cycle occur in and it occur in two pathways. They are C4 pathway. This pathway occur in mesophyll cell and also one more pathway it also follow, that is C3 pathway. So C3 pathway occur in bundle sheath cell. Here note is C3 pathway is a part of C4 cycle. And now coming to C4 cycle occur in 4 steps. C4 cycle occur in 4 steps. They are first one carboxylation or also carbon dioxide fixation, and the second step is reduction, and the third step D, and the fourth step is re of TP means phospho. First two steps like carboxylation and reduction, these two steps occur in mesophyll cell and they follow C4 pathway. And last two steps like decarboxylation and regeneration of PEP occur in bundle sheath cell and they follow C3 pathway. And here in which C4 cycle, in C4 cycle carbon dioxide fixation occur in twice. First carbon dioxide fixation occur in mesophyll cell by PEP. And it is a 3 carbon compound in presence of enzyme called PEP carboxylase enzyme, and to continue C4 pathway. And second carbon dioxide fixation occur in bundle sheath cell by RU, and it is a 5 carbon compound. RUBP means ribulose 1,5 bis. In presence of this RUBP accept second carbon dioxide in bundle sheath cell in presence of rubisco enzyme. It is the largest and most abundant enzyme in the world. And after in presence of rubisco enzyme, RUBP accept second carbon dioxide in bundle sheath cell to continue C3 pathway. Now let us discuss each steps in details. First step, that is carboxy, also called carbon dioxide fixation. In this process, carbon dioxide accepted by 3 carbon compound called PEP in presence of peptase enzyme to form 4 carbon compound, that is oxaloacetic acid has 4 carbon, PEP has 3 carbon, and in carbon dioxide 1 carbon is there. So due to formation of 4 carbon compound, and it is the first stable photosynthetic product, hence the name came as a C4 cycle. And this oxaloacetic acid is unstable. To become stable, then this oxaloacetic acid undergoes second steps. So that is the reduction steps. In reduction, this unstable oxaloacetic acid 4 carbon compound in presence of ATP energy, and it gives rise in presence of malic dehydrogenase enzyme, and it become form into malic acid, and it is also a 4 carbon compound, and malic acid is a stable product. And the formation of malic acid occur in reduction process, and the first these two steps occur in mesophyll cell to continue C4 pathway. And after formation of malic acid 4 carbon compound, then it enter into third steps, so that is decarboxylation step. In decarboxylation step, this decarboxylation step occurring in bundle sheath cells. These malic acid 4 carbon compound in presence of NADP plus in presence of an enzyme called NADP malinase enzyme, it converted into 1 carbon in the form of carbon dioxide and 3 carbon compound in the form of pyruvic acid. So pyruvic acid has a 3 carbon, from in carbon dioxide 1 carbon. So this is occur in bundle sheath cells by decarboxylation process. Here the formed carbon dioxide in bundle sheath cell was accepted by 5 carbon compound RUBP in presence of Rubisco enzyme. Then it form 3 carbon compound, so that is 3-PGA, 3-phosphoglyceric acid, and the formation of this 3-phosphoglyceric acid is a photosynthetic stable product in C3 cycle, and from this 3-PGA, then this 3-PGA will continue to C3 cycle. So by end of the C3 cycle, from 3-PGA, there is a formation of 1 molecule of glucose. Here glucose molecule will synthesize in this decarboxylation, and this is what C3 cycle. And the formed pyruvic acid. So this 3 carbon pyruvic acid, which later undergoes 4th steps, that is regeneration of RUBP. So regeneration of PEP. Here from this pyruvic acid back to phosphoenol, and it is also 3 carbon, here also 3 carbon, and this PEP, why this PEP will again regenerate means to continue another cycle to C4 pathway, to continue C4 pathway in C4 cycle. So there is a must and should formation of PEP. So like that, from these 4 steps the C4 cycle will occur. And now coming to most important things here, we need to know that and energy consumptions, energy requirements, total energy requirement for C4 plant. We will discuss total energy requirement for C4 plants. It is very most important. Total energy requirement for C4 plants. Right. For 1 carbon dioxide fixation in C4 pathway, use 2 ATP, no N, but in C3 pathway use 3 ATPs and 2 NAD. So total for 1 carbon dioxide fixation, total 5 ATPs and 2 NAD is required. For example, for 6 carbon dioxide fixation means synthesis of 1 molecule of glucose, because to synthesis of 1 molecule of glucose we need 6 carbon dioxide fixation. For this, in C4 pathway, use 6 into 2 ATPs, so total 18 ATPs, sorry 12 ATPs, and it is the extra ATP in C4 pathway. It is the extra ATP used in C4 pathway to helps to prevent photo process, are also called C2 cycle. And in C4 pathway for 6 carbon dioxide fixation, use no N. Now coming to C3 pathway, for 6 carbon dioxide fixation, use into 3 ATPs equal to 18 ATPs, and it use 6 into 2 NAD, and total 12 NAD will be used. So total there are for 6 carbon dioxide fixation or for the formation or for the synthesis of 1 molecule of glucose, there is a need of 30 ATPs, that is 12 from C4 pathway and 18 from C3 pathway, and 12 NAD is required, and this 12 NADPH2 only from C3 pathway. So it means that no C4 pathway utilize NADPH2. So finally, for synthesis of 1 molecule of glucose, for synthesis of 1 molecule of glucose or for 6 carbon dioxide fixation, we need 30 ATP and plus 12 NAD. For example, for C3 plants, for C3 plant, total ATPs and NADPs are only 18 ATPs are utilized and 12 NADPH2. So for C3 plant, 18 ATPs and 12 NADPH for C4 plants, 30 ATPs and 12 NADPH2 are required for the energy requirement for the C4 plants. Now coming to C4 plant has some characteristic features. Now we will discuss, so what are the important characteristic feature of C4 plants. Generally C4 plants are found in dry tropical region. So now we are discussing C4 plants salient feature. C4 plant salient features. First one, C4 plants are generally found in dry tropical region, and these C4 plants tolerate high temperature. They also tolerate high light intensity. And most important feature of C4 plant is the C4 plant shows branch anatomy in leaves of C4 plants. So what is branch anatomy? So branch is a Greek word in which, not Greek, it is a German word. So here branch means wreath or ring or circular like arrangements of bundle sheath cells around vascular bundle. So here vascular bundle means xylem and phloem. For example, if you see here the structure of leaf, so here we have upper epidermis, here we have lower epidermis, and between these we have vascular bundles. Above we have xylem and phloem. Around xylem and phloem we have some specialized cells. They are called vascular bundle sheath cells, like this. So these are what? All bundle sheath cells. And the arrangement of bundle sheath cells around vascular bundles are like a ring like or wreath like or circular like arrangement. This type of arrangement of bundle sheath cells around vascular bundle is called branch anatomy. And these cells, the bundle sheath cells which appear around vascular bundles are larger in size. These bundle sheath cells around vascular bundles are larger in size. So this type of special arrangement of these cells around vascular bundle is called branch anatomy. Now these C4 plant also shows chloroplast dimorphism. A dimorphism means two structures, like chloroplast has a two structure. It means that generally chloroplast in mesophyll cells, in mesophyll cell, a chloroplast is normal chloroplast, and with the chloroplast has grana, and they contain with grana, and it carry these mesophyll cell or normal chloroplast carry C4 pathway. But when we see the chloroplast in bundle sheath cell, the cell is abnormal. Because of these abnormal cell or bundle sheath cells are more larger than any normal cells, and their chloroplast is abnormal chloroplast. They contain abnormal chloroplast. Here abnormal chloroplast means a chloroplast without grana, okay, and here there is no gases exchange will occur in this abnormal chloro, and these bundle sheath cells abnormal chloroplast carry C3 pathway. It carry C3 pathway. And one more important features of C4 plant is no photorespiration. Here, sixth character here I am writing. So it doesn't carry photorespiration. No photorespiration or C2 cycle means, and C4 plants prevent photo process. We will discuss in next upcoming videos what is photorespiration, what is the importance of photorespiration. We will discuss, and we will make another videos for this special topic. Okay, it prevent photorespiration. So due to prevent, hence no carbon dioxide, no loss of carbon dioxide. Due to prevent of photorespiration by this C4 plant, no loss of carbon dioxide occur, and no yield will be lost, and high productivity. So we can, these C4 plants obtain high productivity. So prevent the photorespiration process, so we can get high productivity by preventing them from loss of carbon dioxide, so that we can yield more yield and we can get high productivity. So this is all about the C4 cycle. Thank you.